Now, let us talk about the problem which we are focused on in this particular course, and that is your EEG signal, which is electroencephalogram, right. So, what is EEG? Now, EEG or electroencephalogram is commonly used in medical and research areas to record the biopotentials from brain, placing electrodes on the scalp, on the surface of the scalp. What exactly we get from EEG, or how it is measured, or how it is generated. So, it is generated because of the electrical activity of the brain. And what does it show? It gives a high temporal resolution for milliseconds and low spatial resolution. Who was the first one? Waldemar 1912, he recorded EEG from animal model, which is dog. Cybulski in 1914 first recorded EEG signals of induced seizures. Seizures mean nothing but epilepsy. We will discuss this in detail. While Berger in 1924 recorded first EEG signal from human and coined the term electroencephalogram. This is some history about the EEG. So, we discussed in previous modules, right, what are these FZ, frontal lobes and occipital lobes and parietal lobes, right, and temporal lobes. We discussed about that. We also discussed about the 10-20 system. So, that there is a uniformity in the placing of the electrodes immaterial of the size of the brain or the head size. So, EEG is a single neuron activity produce two small signal to record, or how EEG is measured. EEG is a sum of the signals generated by multiple neurons, because single neuron activity produces two small signal. Second, EEG reflects the summation, as I said, summation of the synchronous activity of many neurons with similar spatial orientations. It is difficult to detect signals from deep sources, subcortical areas, than the areas near the skull. Okay. So, if you see a single neuron, what you will see? You will see dendrites, then you will see neural body, you will see axon, and you will see sympathetic terminals. While the synapse, right, it is connected with the postsynaptic membrane. Alright. So, you can see the signals like this. We will take in a new slide and we will understand further. You can see here the 10-20 system, how it is connected. So, how the EEG signal is originated, alright. So, if you see here this particular schematic, there is a afferent axon and there is a efferent axon, right. The effective synapses creates a potential, and the group of neurons, right, in a synchronized manner, the signal is transmitted through the pia mater, right, to the suprainchoate space, arachnoid space, dura mater, all the way through skull to the scalp and to your EEG electrode. Alright, this is the group activity, and you can see the active synapses, how they generate a potential. So, in another way, if you want to see further, so if this is the EEG electrode, right, and this is the same schematic which is here, but of a different size. Then if I take this the neurons, then you can, if you number it 1, 2, 3, 4, 5, 6, alright, and then we want to measure the signal from each neuron. You can see there is a irregular pattern and there is a synchronized pattern, and EEG is nothing but summation of these patterns, the summation of the signals generating out of group of neurons, right. So, in the synchronized manner, you can see that it is a clear summation, we can see the change in the activity. So, many neurons need to sum their activity in order to be detected by EEG electrodes. The timing of their activity is crucial. Synchronized neural activity produces large signal. You can see here, compared to the irregular EEG signal, when you sum it and when you take synchronized EEG signal and you sum it, the synchronized version produces, or neural activity produces a large signal. So, now we understand that we by placing an electrode on the scalp we can measure the brain waves. So, what are brain waves? Brain waves are commonly measured as peak to peak voltage and normally range from 0.5 to 100 microvolts in amplitude, which is approximately 100 times lower than ECG signal. You can see here, right. It is only few microvolts, where in ECG it is about few millivolts. So, at least 100 times lower than EEG signal, and that is why the difficulty will be when you measure this signal, because you have to take care of the noise interfering with your signal, alright. So, that is one thing. What we can do, using Fourier transform power spectrum, the raw EEG signal is derived. The brain state of the individual may make certain frequencies more dominant, alright. So, while brain waves have been categorized into 4 basic groups. So, depending on the individual, the brain state would be different, and certain frequencies would be more dominant compared to others. So, what are these frequency ranges? So, based on the brain waves are basically divided into 4 groups, and those 4 groups are beta, alpha, theta and delta. Now, let us see each of those. The beta is the awake, non-focused, relaxed, drowsy or non-vigilant low level of environment stimulations. It is around greater than 13 hertz, and you can see the beta right over here, right. It looks like this. So, when you are not dead, you are awake but non focused, and you are relaxed and drowsy, and you are non vigilant, and then at that and low level of environmental stimulation is there, at that time beta waves can be measured using your EEG. EEG will show beta waves. Alpha, it is 8 to 13 hertz, this is the alpha signal. You can see a pattern 8 to 13 hertz, and these signals are awake, focused, attention, problem solving, dream, REM sleep, high level of environmental stimulation. Example, eyes are open, you are talking, you are moving your eyes, you are in the REM sleep, or you are focused and attention. At that time your alphas signals would be there, while the theta is 4 to 8 hertz. Theta is 4 to 8 hertz, and visual imaginary, right. And you have a light sleep, or you are on a hypnopompic imagery. At that time you will be able to see the theta waves. Finally, the delta, which is between 0.5 and 4 hertz, shows a deep restful sleep, wake dream states. This is the delta, alright. So, brain waves are divided into 4 groups. You can say delta, theta, alpha, beta. Delta, deep restful sleep, theta, visual imaginary, alpha, awake, focused, alert, beta, non focused, relaxed, drowsy, alright. And this is the signal, EEG signal, when the person is suffering from epilepsy, alright. So, there are these are the brain waves that we can divide into groups. So, now let us see, EEG potential are good indicators of global brain state. They often display rhythmic patterns at characteristic frequency. You can see excited, it will look like this, relaxed, right, then drowsy, asleep, deep sleep and coma, right. All this frequency we can measure, or the patterns we can measure depending on the state of the brain, right. Same thing is here again, right, gamma, beta, alpha, theta and delta. A gamma frequency is there, which is between 31 and 100 hertz also, and it shows a peak focus, expanded consciousness. It is very difficult to see the gamma frequency in human brain waves. We require a deep focus, insight and a really expanded consciousness to see the gamma, while beta is alertness, concentration, cognition, alpha relax, theta meditation. When you do meditation, theta waves are there, and delta is a detached awareness, healing, sleep, right. These are the states of the brain waves. Now, There are different procedure of EEG recording, and like I said, if you divide it from the Nasion, right, you will say 10, 20, 20, 20, 20 and 10, and you read to Inyon. Alright, from Nasion to Inyon, this is how it is divided. I told you that on the right side it will be even number, and on the left side it will be odd numbers. So from Nasion is this part, and Inyon is this part. Okay, so in the another viewpoint you can see the things, and that there is a pre-aureal point, right, which is right over here. So if you say vertex and you take it, then this is somewhere your pre-aureal point. So standard EEG makes use of 21 electrodes linked in various ways or montages, we can say. Apply electrode according to 10-20 system, check the impedance of the electrodes. Impedance is very important, and to reduce the impedance in wet electrodes we generally use the gel. For dry electrodes impedance is already low. So if you measure continuous EEG recording, from the EEG recording we can understand if there is a problem with the brain waves, particularly if you want to see kind of epilepsy and other phenomenon. The EEG recordings are done. You can see the recordings from all these electrodes, and of course, you already know F stands for frontal, T for temporal, C for central, O for occipital, right, and it is an international 10-20 system. So, what are the advantages of EEG and what are the disadvantages of EEG? The first advantage of EEG would be good time resolution, that is in millisecond, compared to compare with seconds, which will be in fMRI. So, what is fMRI? fMRI is a functional magnetic resonance imaging or functional MRI, and what it measures? It relies on the fact that cerebral blood flow and neuronal activations are coupled. Okay. So, when the area of brain is in use, blood flow to that region also increases. This is the principle of fMRI. So, it is a relation between the cerebral blood flow. Cerebral blood flow is a blood flow in the cerebra, and neuronal activities. So, if more the blood flow, that means activity is more, and activity is more, there is more blood flow. So, this is how it is calculated, but fMRI is of few seconds, while in EEG it is only milliseconds. Second is EEG system is portable and affordable. In fMRI is difficult, more tolerant to subject movement compared to fMRI. EEG is silent and so useful for studying auditory processing, can be combined with fMRI or TMS. The difficulties or disadvantages or limitations of EEG are low spatial resolution, and the artifacts and noise will cause the distortion in the signal. So, you should have a conditioning, electronic conditioning modules which are of very high signal to noise ratio and good quality. So, let us see 3 videos now, right, for the EEG and brain waves. I will play one by one.
When you arrive for your ERP session, you will be greeted by Frank and Nate. You can play with Nate while your family talks with Frank and reads about the study.
It was great to see that Charlie was comfortable playing so that I was able to talk to Frank and read about the study.
While you play, Nate will show you the caps that you will wear during the study. They are stretchy and have lots of little holes in them. He will show you the syringe that he will use to help him get the cap ready. It looks like something that you might get a shot with, but it's not sharp at all and it won't hurt you. It's only used to put some sticky gel into the holes in the cap. You'll be able to put a cap on yourself and play with the syringe. That way you will know exactly what will happen when Nate puts the cap on you.
Nate let him touch the syringe so he would know that it wasn't sharp or scary. It also helped prepare him for seeing it again later when it was his turn.
When playtime is done, you'll sit in a big white chair and pick out some cartoons you'd like to watch. You'll get to watch the first cartoon while Nate gets you ready for your cap. First, he'll have to clean around your eyes and behind your ears with a wet cloth. Next, he'll brush your hair and measure your head to be sure exactly which cap will fit you best.
It was nice to get to sit right there with him so that I could see what was going on and be there if he needed anything.
Before he puts the cap on your head, he'll put a few wires on your face with some stickers. When he's done with the stickers, he'll put the cap on your head.
While they were putting the cap on, they explained the different steps so that nothing came as a surprise.
Then, using the syringe he showed you during playtime, he'll put some cold gel in each one of the holes in the cap. After there's gel in all of the holes, you'll go into the little room to watch a video that you selected earlier. You can ask your mom or dad to come with you into the little room if you'd like.
I got to go into the sound booth with him, which helped him feel relaxed. I thought it would be hard to help him stay still, but the room was calm and quiet, and we just sat and watched the cartoons.
Inside the study chamber, it's dark and quiet, and you'll get to watch another cartoon. While you're watching this cartoon, it's important to be very still and very quiet. After you're done watching the cartoon, the cap and stickers will be taken off.
They took the cap and stickers off and told him what a great job he did.
When that's done, you'll get to play and have fun in the lab spaceship.
Charlie thought all the equipment was cool, but I think his favorite part was getting to climb into the spaceship at the end. Thanks to their patience and care in explaining the process, it was a great experience.
Backyard Brains presents the electroencephalogram, also known as the EEG. Observe the alpha waves of the visual cortex of the human brain. For this experiment, you need a heart and brain spiker shield, an Arduino, a computer, and yourself. To begin, take the Backyard Brains custom EEG headband and place it on your forehead such that the electrodes on the back of the headband are located on the back of your head. For the ground, we will place an adhesive electrode on the mastoid process, which is the bony projection you can feel behind your ear. To improve the interface, we will place some conductive gel in between the electrode and the skin. If you have long hair, you can part the hair directly underneath each electrode to further improve the quality of the signal. With your orange interface cable, attach the red alligator clips to the electrodes on the back of the head, which is which does not matter, and the black alligator clip to the ground behind the ear. We then plug the orange cable into the orange port on the heart and brain spiker shield and the USB cable on the other side. The other end of the USB cable goes into the computer, and we are ready to begin. But what is going on here? When your eyes are open, the visual cortex in your brain is processing a lot of information about your entire visual field, about levels of contrast, color, and light. But when your eyes are closed, the field is dark, and hence, counterintuitively, the neurons in your visual cortex become more synchronized. A popular analogy is to imagine yourself outside a stadium during halftime. There are a lot of conversations occurring inside the stadium, a lot of information processing. But outside the stadium, all you hear is a dim hum of noise. This is equivalent to the eyes open condition in the electroencephalogram. Alternatively, during the singing of the national anthem, many of the spectators inside the stadium are singing the same thing. They are synchronized, and this signal is strong enough that you can plainly hear it, though distorted, outside of the stadium. This is equivalent to the eyes closed condition of the electroencephalogram. The physiological underpinnings of the EEG signal are complex and still a topic of active investigation in the neuroscience community, but we currently understand it to be the summed activity of many synapses in the upper layers of the cerebral cortex. Our heart and brain shield thus amplifies the electrical activity of these synapses such that we can view them on a computer under the appropriate conditions. Click on the settings button in our Spike Recorder software and select Connect via USB port. Within a couple seconds, you should see the signal change to a putative EEG signal. You can zoom out of the timescale by using the two-finger motion on your trackpad or the scroll wheel on your mouse, and zoom in on the y-axis by clicking on the positive button on the left side of the screen. But how do we know this EEG signal is real? Well, let's close our eyes. Those ripples are the 8 to 10 Hz alpha waves of the visual cortex that disappear when the eyes open again. If you click on the FFT button, you will bring up a spectrogram view, which will show the EEG signal decomposed into frequency, time, and amplitude. Thus, when the eyes are closed, you will note the increased signal strength at 8 to 10 Hz. Under appropriate conditions, the alpha waves of the visual cortex are readily apparent, whether you're showing them to your friends on a Friday night in your living room or in front of 200 people for your high school or college lecture course. These alpha waves were first discovered by Hans Berger, a German physiologist in the 1920s, and subsequently verified by Lord Edgar Adrian at the University of Cambridge in the 1930s. We thank our many friends at the Santiago Makerspace in Chile who worked with us to replicate these findings in a compelling and simple way. We also thank our production team, and this is just the beginning as we further our explorations into the EEG signal. What will you discover? Backyard Brains, neuroscience for everyone.
So we're going to be recording your EEG today, and so essentially what we're going to do is put this swim cap over the top of your head. It doesn't have any feeling associated with it or any encumbers. It's just a pretty easy process.
Okay.
Put these little foam sponges on these here. And now the cap will go over your head. Okay. This is the hardest part of the whole process is right here. This cap goes right over the top of your head, just above your eyes. So. Just like that. And you get two ear sensors here, one on each side. Okay. And on the other side over here. Okay, so the hard part's over. So now from here, all we do is I have this syringe. It looks like a needle, but it's not a needle, just so you can see it's not sharp or anything here. And so what I do is I take this and I put the paste into each of these sensors here. So here, just like that. And one in each of these holes so that we can get a good connection between the sensors and your scalp. So there shouldn't be any pain involved or anything like that. It's just connection between your sensors and your scalp. So that's all there is to it. And so what you'll do now is just sit here for 10 minutes with your eyes closed. You look forward, sit nice and still, relax your shoulders, your jaw, your forehead, all that. And I'm going to be behind you here recording the EEG. We'll do that for 10 minutes with your eyes closed, then we do 10 minutes with your eyes open, and then we're done. And that's all there is to it.
Okay, so you have seen 3 videos of the EEG, and from there what you see is that how EEG signals, how the electrodes are placed, what are the EEG signals, what are the ways to acquire this EEG recordings, right. But let us also see what are the applications of EEG, and we will finish the module at that point, and then we will continue about the electronic conditioning circuit in a later module. So this is the last slide of this module, and this is on the potential applications of EEG signal. So, what are the potential applications of EEG signal? The first application is to understand monitor alertness, coma and brain death, because from the brain waves we can understand whether the brain is dead or the person is in coma or there is a alertness is there or not. Second is we can understand areas of damage following head injury, stroke or tumor. We can also test afferent pathways by evoke potentials, monitor cognitive engagement, alpha rhythm, produce biofeedback stimulations, control anesthesia depth, which is controlled by servo anesthesias, in another term it is called servo anesthesia. Investigate epilepsy and local seizure, we will discuss about it in detail. Test epilepsy drug effects, whether a drug is having a high efficacy or not, we can understand by measuring the EEG signals or ECOG signals in some time or for some applications. Assist in experimental cortical excisions and epileptic focus, monitor human and animal brain development, test drugs for convulsive effects, and finally investigate sleep disorder and physiology. So, there are many applications of EEG because finally EEG is related to nothing but your brain waves, and brain waves shows a lot of interesting applications right from the deep sleep to a person whether he is in coma or not, to a person whether he is attentive or not. Let me give an example. If you can make a band to understand whether the person is attentive or not, then you will know the productivity of an employee. Suppose I wear a band, I work for 12 hours, how productive, how effective I am while I am working, because your brain waves will show that okay, your attention is decreasing and you can take a break in between, right. Once you take a break, when you come back again the signals will be different. That means you can measure the attentiveness of a person and you can improve the quality of the time invested by an employee in an organization. I am just giving an example. Okay. Same thing you can also use for truck drivers. If they wear the band and as soon as you see that they are sleepy or they are less attentive, you can alert them on the dashboard saying that take a break, right. This is another example. So, if you understand the brain waves, if you understand EEG in detail, you can use for thousands of applications in the area of research. And that was the idea of this introductory, you understand the name of the course, Introductory Neuroscience and Neuroinstrumentation. Alright, we can take an example and go in depth of each of those maybe in the next course, where it will be advanced level of neuroscience and neuroinstrumentation. However, we will cover lot of instrumentation techniques in this course, and like I said, we will be discussing about the clean room environment. We will be showing you some experiments within the clean room of how to operate the equipment. We will also show you how to use the EEG electrodes and how to acquire signals, and of course a lot of things on neuroscience. Alright. So, I will conclude this class here, and I will see in the next module talking more about the electronic modules for EEG acquisition. Till then you take care, I will see you in the next class.